Thanks everyone for coming. I'm sure you're grabbing a last-minute coffee. For those of you that are jet lagged, that'll help a little bit, but when you see my talk, it doesn't really do you that much good in the end. Before we get started, I want to say a few words. As some of you may know, the final speaker of our session today was supposed to be Dr. Thomas Connor. Sadly, Thomas passed away in February. My fellow speakers, along with the Society, have thought it appropriate to dedicate the session to Thomas's memory. Dr. Andrew Harkin, one of Thomas's long-term colleagues at Trinity, has graciously agreed to give a talk in Thomas's stead, and I'm sure we'll hear about some of the fascinating work that group has been doing. With that, I'd like to introduce our first symposium speaker. I promised him I wouldn't embarrass him, but I did include one date, Bruce. So, Bruce is the Alfred Mirsky Professor at the Rockefeller University. He got his PhD from Rockefeller in 1964, 14 years before I was even born. He is a member of the National Academies, the Institute of Medicine, and a fellow of the American Association for the Arts and Sciences. He's also past president of the Society for Neuroscience. He has won numerous awards, including the Ipsen Foundation Prize in Neuroplasticity and the Carl Spencer Lashley Award from the American Philosophical Society, to name just a few. This is the shocking statistic, but he has also been author on over 1,000 articles, reviews, book chapters, and authored several books. So, yeah, it's a tough act to follow. He also happens to be my postdoc mentor, and I can safely say it was a remarkable experience. Bruce has been a luminary in understanding how steroid hormones and stress can affect neural circuits and behavior, which is currently active in my brain, and as such, I think serves as a perfect lead-off speaker to our symposium. The title of his presentation is Good Stress, Bad Stress, and the Effects on Neural and Behavioral Function. Bruce?
Thank you, Ilya, and good afternoon, all you mostly young whippersnappers. I want to talk to you about stress and sort of introduce the topic of the rest of the symposium and start out by reminding you, of course, which we all know, that when we think of stress, we often think about cortisol, and cortisol, as we know, is directed from secretions from the hypothalamus to the pituitary, and when it's secreted acutely, it actually has beneficial effects, even though it's often talked about in the context of bad events, but emphasizing that Mother Nature put it there in order to help us adapt and survive. So it enhances memory, enhances immune function, promotes energy replenishment, and more efficient cardiovascular function. But we know that chronic stress and elevated levels of cortisol, glucocorticoids, can have immunosuppressive, anti-inflammatory effects, impair memory, promote bone mineral loss, muscle wasting, and metabolic syndrome. And since the immune effects are perhaps often overlooked, work of a former PhD student, Dr. Firdaus Dabar, now at Stanford, showed a number of years ago, using delayed type hypersensitivity, sort of like a tuberculin skin test, that the application of the antigen to which there is an immune memory enhances the flow of immune cells measured by the thickness of the ear. And if one stresses the animal acutely, a moderate stress or a more severe stress, there is an enhancement of this response, which is actually mediated both by catecholamines and by cortisol in a very complex and interesting way, as well as local mediators that are sort of chemotactic. But that's what happens with acute stress. If one does chronic stress for a period of, in this case, restraint for a period of 21 days, there is a profound suppression. You'll notice the y-axis is different. There's a profound suppression of DTH. So there is this biphasic effect. And you saw on the previous slide the fact that there is an inverted U, which is a description of many of what happens with many of the mediators of stress and adaptation. And this diagram points out that cortisol is only one of many mediators, and there are many more, including metabolic hormones, that they all talk to each other. There is nonlinearity, and they affect systems throughout the body. And this is particularly important since we tend, especially in medicine and to some extent in biology, with the old schism which is changing between the brain and the rest of the body, to operate in silos. And thinking about these mediators and their simultaneous effects on many systems of the body as well as the brain is a very important thing to keep in mind. A number of years ago, Peter Sterling and Joe Iyer at Penn described this concept of allostasis, which is the active process of putting out mediators like adrenaline and cortisol that leads to adaptation. And in the context of cardiovascular disease and cardiovascular function, they talked about the elevation of heart rate and blood pressure that occurs when you stand up, walk up a flight of stairs, give a talk, have an argument, and so on. And they noted, of course, that if this basal level of heart rate and blood pressure is elevated, it can go up further when one does one of these common things, but it leads then to, over time, can lead over time to pathophysiology. And with the late Elliot Steller, also at Penn, we renamed this long-term effect allostatic load, or the cost that the body pays for doing its job when the system is overused or dysregulated. And the metaphor is a seesaw that remains in balance. Homeostasis has remained, but there are two heavy weights that over time can cause pathophysiology and damage. But then we skip to the natural world, and sometimes I think that this means that we humans live in an unnatural world, which may be increasingly the case. But if we look at the natural world and the pioneering work of John Wingfield, talking about birds that encounter a sudden storm or flood in the spring when they're getting ready to mate, the role of the cortisol elevation is, as summarized here, to suppress unnecessary physiologic activity such as reproduction, to delay it at least, to mobilize energy reserves, and to activate alternate behavior patterns such as getting out of there, going to a safer place. And then we can think about something which we had called allostatic load. And to this, I'm grateful for a collaboration with John Wingfield, thinking about this in, again, the natural world, looking at what happens to animals that are preparing to hibernate, putting on fat for the winter, which if these bears put on this fat living in a zoo might lead to cardiovascular disease and diabetes, as it does sometimes in humans. But here it's a natural process because the animals burn it off during the winter and then resume their activity when the weather conditions improve. And then there is the migrating salmon, which essentially dies of an allostatic overload. The term overload was suggested by John to emphasize the the most negative effects, but in this case, it's a positive for the environment because it frees up resources and provides food for the next generation. And then we come then to the transition from the natural world to the unnatural world that we live in, where Robert Sapolsky's wonderful book, Why Zebras Don't Get Ulcers, presumably they don't get ulcers because they don't worry, but then for those at the back, this is a cartoon from the New Yorker. These are the Fortnoys. The Fortnoys are the young worrieds, and living in New York, there is this poster in one of the subway stations, and I think it highlights the fact that we often do ourselves harm by worrying too much about things, but indeed we do have many things in our busy lives that can be stressful. So getting back to this concept of allostasis leading to adaptation and allostatic load as the price, if we think about the behavioral responses to being in stressful situations. We can think of becoming stressed out, which I think we all experience from time to time, feeling overwhelmed, out of control, exhausted, anxious, frustrated, angry. And what happens to us? We may lose sleep at night. We may eat too many of the wrong things, drink alcohol, smoke if we're so inclined, and neglect regular moderate exercise while we're trying to finish that paper, read that paper, finish writing a paper, and answer all those email messages. All of these contribute to allostatic overload, so psychosocial stress is a major factor, in addition to, incorporating the lifestyle that results from that. In a study with the MacArthur Foundation Network on Socioeconomic Status and Health, Teresa Seaman at UCLA has pioneered in the use of common measures collected often in health exams to develop a composite score in which each of these, if one is in the extreme quartile, counts as a score, and you can have, I think, up to 14 or 17, a score of up to 14 or 17 in this very simple scoring system. And what this scoring system has done is to predict mortality in a successful aging study over 7 years time span, to relate higher education to lower allostatic load scores, but African Americans have higher allostatic load scores and a flatter gradient across socioeconomic status. Neighborhood poverty, not surprisingly, and neighborhood chaos, higher allostatic load scores, social conflict, also higher scores, but social support, lower scores. So it's become a fairly useful measure in epidemiology, public health, and now possibly in the clinical applications. And next we come to the brain, which is our area of primary interest, and what happens to the brain as a result of acute and chronic stress. I'm going to talk briefly about 3 areas. First, the hippocampus, then the amygdala, and the prefrontal cortex. We know the hippocampus is involved in spatial memory, also mood regulation, but it also helps to shut off the HPA stress response. The amygdala, of course, anxiety, fear, and aggression, helps to turn on the hormonal and autonomic stress response. And the prefrontal cortex, which is important for various self-regulatory behaviors, for decision-making, working memory, impulse regulation, mood regulation, and it also helps to shut off the physiological stress response. So what happens to the circuitry of these 3 brain regions as a result of stress is very important for how these brain regions interact with each other and how they perform their functions. And we know now that in the adult as well as developing nervous system, there is continual remodeling of dendrites, at least of some neurons. Dendrites get longer, they can get shorter and less simplified, less branched. That some excitatory spine synapses are turning over all the time, and that there is a limited amount of neurogenesis, particularly in the dentate gyrus region of the hippocampal formation. And all of these are regulated by experience, aided in many cases by hormones, by a whole host of intra- and extracellular mediators. In studies on the hippocampus, having discovered that the hippocampus has receptors for adrenal steroids, and looking at this trisynaptic circuit, looking in particular at the CA3 region, which receives input from the dentate gyrus, is very important for the memory of events in space and time, and also because of its circuitry is vulnerable to excitotoxic, as in seizure-induced damage. With repeated stress, the 3 weeks of stress that cause suppression of delayed type hypersensitivity. If you remember that earlier slide, there is debranching of the apical dendrites of the CA3 neurons, and the basal dendrites do not appear to change. This is mediated by the release of excitatory amino acids, which are elevated during acute restraint stress, but here you see that adrenalectomized animals do not show that elevation of glutamate, suggesting some interaction with circulating adrenal hormones. And indeed, blocking glucocorticoid synthesis while stressing the animal prevents this remodeling. Likewise, blocking NMDA receptors and blocking excitatory pathways with lithium or delantin, also with benzodiazepines and certain antidepressants, as well as a deficiency of BDNF leads to an impairment of this, what we call adaptive plasticity. Neurogenesis is suppressed. It took, however, as much as 6 weeks of daily restraint. 3 weeks is sufficient to cause dendritic remodeling, but 6 weeks produced a significant reduction not only in neurogenesis but also in the volume and the number of granule cells in the dentate gyrus of the hippocampal formation. So we can actually shrink the hippocampus with chronic stress, and others have shown that the hippocampus can be elevated in volume by regular exercise. In 3 different animal models, in collaborative studies with the Blanchards, we showed that the visible burrow, both the dominants as well as the subordinates, showed CA3 dendritic remodeling, with the dominants showing surprisingly somewhat more than the subordinates compared to cage control animals. With Eberhard-Fuchs, we showed that in the tree shrew, the intruder under chronic stress shows dendritic remodeling, also suppression of neurogenesis. In the hibernation model with Paul Privé, we showed a very rapid onset of dendritic shrinkage and a rapid reversal when the animal was awakened from torpor in a matter of hours that caused dendritic regrowth, which suggests that this is cytoskeletal remodeling and doesn't involve significant amounts of, say, protein synthesis and so forth, at least to reestablish the dendritic tree. In some ongoing studies by Jason Gray, Carla Nasca, and others in the lab, we've looked at mice subjected to an acute swim stress, naive animals, then animals that have been chronically restrained and subjected to the acute swim stress, or animals that have been chronically restrained and allowed to recover and then subjected to acute swim stress, and compared patterns of gene expression. And what we see is that there is a core of genes going up or down that respond to the acute stress under all 3 conditions, but you can see that there are significant differences in the genes that are turned on and turned off under each of these conditions, showing very vividly that the brain is not the same, and that when we talk about an animal recovering and showing a reversal of dendritic shrinkage after recovering from chronic stress, we're talking about a brain, at least a hippocampus, that is significantly different in terms of genes that are turned on and turned off. So we're also interested in modification of histones and the part of what is now referred to as epigenetic regulation. And I'll give just one example. We have acetylation, which generally enhances gene expression, methylation, which suppresses it. And one example from work of Richard Hunter in the lab is that an acute restraint stress causes a huge increase in the trimethylation of histone H3 on lysine 9. And you can see this dramatically here. And what he found using chromosome immunoprecipitation and then analyzing the DNA that was trapped and repressed is that there were certain so-called retrotransposon-like elements. These are DNAs that either code for RNAs, and those RNAs were suppressed by as a result of the trapping of this DNA, and other kinds of retrotransposons that can rearrange themselves and lead to genomic instability. So it looks like an acute stress increases genomic stability through this mechanism. But with the repetition of this stress, that is after the 21st day, the same acute stress fails to show this increased repression, suggesting that now the genome may have potential for instability. What this means, both good and bad, is unknown, but it certainly brings us back to this inverted U, in which we see that under acute conditions with physiologic activation, there's enhancement of learning, excitability, and so on, but with higher levels of acute stress, the opposite can happen, lasting, however, short periods of time: suppression of LTP, enhancement of long-term depression, impairment of certain kinds of memory. With a longer chronic stress, as I've showed you, we have what we call adaptive plasticity with neurogenesis and dendritic remodeling, and then with severe head trauma, seizures, stroke, as Robert Sapolsky showed very clearly, glucocorticoids and excitatory amino acids interact with each other to actually cause permanent irreversible damage. And finally, we have this condition in the middle where there are, under some circumstances, a loss of recovery, and this may happen in anxiety and depressive disorders. It appears to happen with aging. The brain gets stuck in a certain state as a result of stress and needs some kind of external intervention to reverse at least the morphologic changes. Translating this to humans, others have shown that there is hippocampal atrophy in major depression, in type 2 diabetes, in post-traumatic stress disorder, in Cushing's disease, where there's some evidence that treatment of the Cushing's will cause at least partial reversal of hippocampal volume loss, impaired memory, and better improved mood. However, even in the absence of disease, there is evidence for hippocampal shrinkage in chronic stress, with chronic jet lag, with a lack of exercise, and also with elevated systemic inflammation, which is a common denominator of many of the diseases of modern life. And on the positive side, there is evidence that even in elderly people, regular exercise will increase hippocampal volume, in part probably by increasing neurogenesis. Intense learning will increase hippocampal volume, and antidepressant treatment, some limited evidence for this, but among the best antidepressants is regular exercise. Let's move on then very briefly to other parts of the brain. While the hippocampus shows the shrinkage of dendrites and loss of synapses, spine synapses, so does the prefrontal cortex, at least the medial prefrontal cortex, while the basolateral amygdala from the work of Shona Chatterjee and colleagues in Bangalore shows expansion of dendrites and increased levels of synapses, and so does the orbital frontal cortex, which is functionally very tightly connected to the amygdala in terms of the saliency of reward or punishment. In a recent study with the Chatterjee group, using a single, for the first time in the animal's life, restraint stress or immobilization stress, that's pretty traumatic, because after a 10-day delay, but not before, there's a significant increase in anxiety-like behavior and an elevated plus maze, and after 10 days, there's an increase in spine synapses on basolateral amygdala neurons, but no expansion of dendrites. That takes repeated immobilization stress. But with that delayed increase, we discovered quite by accident, using saline injections, that we could block this. If we stress the animal with a saline injection or by elevating glucocorticoids just before the animal went into this. So here, glucocorticoids have a positive protective effect, as they do in case of inflammation. There's epidemiologic evidence from a clinical study by Gustav Schelling in people undergoing open heart surgery that if they have low normal levels of cortisol, elevating them during surgery will protect against the development of PTSD symptoms. And in an Israeli study with another animal model, but also with people who have experienced a car accident within an hour are given supplemental glucocorticoids, there appears to be a reduction in PTSD symptoms. So this may actually have some relevance. Finally, just to summarize, in an ongoing collaboration with John Morrison's lab at Mount Sinai School of Medicine, looking at this dendritic remodeling in the medial prefrontal cortex. This happens, this is reversible in young adults, but this resilience is lost with aging, even by middle-aged. In a rat, there's only partial recovery in a 3-week span after the stress is turned off. You'll hear from Ilya about circadian disruption. And very importantly is that female rats respond quite differently from male rats in this remodeling effect. And then we come back to the glucocorticoids, which we used to think were exclusively working through the cell nucleus. They still do, both directly through these glucocorticoid receptors and indirectly through interaction with other second messenger systems and their modulators like Krebs and so forth at the genomic level. But glucocorticoid receptors are also translated into mitochondria and regulate calcium balance. Glucocorticoid or adrenal steroid receptors are associated with the membrane in a way in which they can stimulate the release of glutamate in particular, but at the same time they stimulate the production of endocannabinoids, as many of you know, that actually inhibit the release of either glutamate or GABA, depending on the specialization in terms of CB1 receptors of particular terminals. So there is a whole host of interesting and sometimes counterbalancing effects of glucocorticoids interacting with many of these other mediators. And I wanted to call your attention in the last slide to this new paper by Connor Liston, a former student who did some of the pioneering work on the prefrontal cortex, but now on his own, teamed up with Wenbiao Gan at NYU, has shown that disrupting the ultradian oscillation of glucocorticoids with a tiny dose of dexamethasone will stop this glucocorticoid oscillation and will stop the ongoing turnover of a subclass of excitatory spine synapses. And in this paper, showing that in fact this is very important. Both the formation of spines and the pruning of old spines is very important to promote motor learning. And I suggest that if you're interested, you might look up that paper in Nature Neuroscience. And with that, I'll acknowledge the people, at least some of the people over many years who've contributed to this, and thank you, and I'll be glad to answer questions if there's time. Ilya. Thanks.
Thank you very much. I feel like a talk show host here.
This was very nice. Thank you very much. I have a very specific question to the histone modifications, acetylation you mentioned after chronic stress, long-term stress, and I wonder which target gene was particularly affected.
Very good question. I'll suggest you might take a look at a recent PNAS paper by the first author is Carla Nasca. This was her PhD thesis in Italy She's now a postdoc in our lab, and one of the key players is mGlu2-3, the metabotropic, which seems she looked at two different depression models, one mouse, one rat, one environmentally induced, the other a genetic predisposition, and showed that a novel, rapidly acting antidepressant, a natural substance called acetyl L-carnitine, which is present in all of us, actually given exogenously will prevent or reverse the depression, and in particular mGlu2 and a particular acetylation of, I think it's histone H3 lysine 27. And ongoing work in our lab right now, she's showing that acetylcarnitine, apparently working through that same mechanism, will block the effects of stress that I was describing here. So it's an unfolding story, but that's the one connection. There undoubtedly are others.